this working? This is working. All right, everybody. All right. It's been a while since I have updated some of the free software tools you can use to analyze your own data. So I thought I'd make a new one because several new tools have come out at ASMS this year, which is the main mass spec conference, about a month ago. So instead of making one like terribly long, like 5-hour video, which I tend to do, I think I'll break it up into several videos. So we're going to do the new version of Fragpipe and then the new version of dian. And they both have new skyline integration. So we'll go over that too. And so let's get to it. And we'll use some of the data that I generated for a normalization video, which you can find a link to in the comment section, where to download it so you can follow along. Okay. So we're first going to look at the new version of Fragpipe, which is an open source, well, not really open source, but a free software for academics that you can use to analyze your data. Fragpipe. You can see if I can do it correctly. So all you have to do is Google search Fragpipe, and you will see a nice link at the top that's download it. So you click download. We're going to be looking at Fragpipe 22, and what I like to do is I like to download this one that says GRE 22.0 contains the Java 1 run type for Windows. That way you don't have to screw around with Java. So I download that, and it is downloading. And I've already installed this, but let's walk through it a little bit. Okay, so then you click on the folder, unzip it all. Let's see, extract all. Just say, I don't know. I've already done it, so let's just give it another name. Okay. All right, so this should only take a minute. Okay, hopefully it'll speed up. Okay. It helps if you have an SSD drive to do all of this data analysis, so I'd recommend getting one because it speeds up pretty much everything a lot. All right, so anyway, this is extracted, and what I like to do is go to the bin directory, and I don't really know if it makes any difference whether you do the batch file or the little icon here, but let's just try that and see if that works. It will give you a little error on Windows 11. Hit run anyway. And then it found some old workflows from my previous install, so I'm just going to copy those. All right, cool. So once it's downloaded, if you don't have Fragpipe, MS Fragger, IonQuant, and diaTracer, which is the tools that Fragpipe, which is I think the workflow manager basically uses, so you can hit download update. And there you put in your name, email, institution, click all these buttons. And if you're an academic, you could do it. Freely available for academic research, non-commercial or educational purposes under academic license. If you're none of those, then you'll have to pay for it. I think it's kind of expensive, but we don't have to. Okay, so we've already done that. You could download dian, which is the part of the DIA analysis software. It also uses it for some deep learning stuff too, so we can download that. And you can download that there, and I think you just install it and it finds the right directory, or it's actually downloaded. Look at that. So it's actually under the tools. But if you wanted to download more recent version, used built-in version, the file, do not use dianx or installer file. If not customized, use built-in. So we're just going to use that because it's built in. Python, you could download Python. And if you want to generate spectral libraries, you can click on install, upgrade, easy PQP, which I think we'll need, but it already says it's available. So if it's not available, I think you'll have to download that. Okay, so all of that's done. Now all we have to do is go to workflow, and because the data we generally generate in the proteomics core is data independent acquisition. I believe I have a few videos on that. And we're going to be looking at data independent acquisition from timsTOF files. So we are going to use the workflow diaa speclib quant diaPASEF. And we're going to hit load workflow, and it'll say, do you want to? You hit OK. All right. So now we're going to load our raw.d files. These are timsTOF files, and you can download these from our BioShare. But these are the ones we're going to use for this example, and there's 8 of them. And you can just kind of drag them into here. And these are a series of just really dumb HeLa lysates that I ran. Some at 200 nanograms and some at 50 nanograms, because I wanted to make a video on normalization and what the normalization does and how it works. So this will be a good, not great example. So we're going to give this an experiment. We're going to call it 200 nanograms, and you can tell which one's which because it will be in the file name. So HE200, HEL 50, that should be HEL 200, but I did that wrong. So we'll do 50 nanograms. And what I like to do is copy these because I believe these are case sensitive, and so any little spelling mistakes will screw up the conditions. So I'll copy that here and here. And then a trick is you can click on this little tab here, which will sort them. And then you can see all the 200s are at the top. And then I'm just going to give it a replicate. This doesn't really matter. But 1, 2, 3, 4. And then I'm going to set all these as DIA. Boom. All right. So all of the files are in there. They're all set on the condition. Let's see, this will use this new software called diaTracer to do the to take the diaPASEF files and convert them into a format that it can search. And if you want to learn more about diaTracer, you can just go to Google diaTracer, and there it goes. You can pull up the diaTracer information. And I believe there is a publication in bioRxiv that you can open and learn all about it. I would recommend reading it if you want. I've read it. It's pretty cool. Okay, so that's all set. We're going to leave everything the same. These HeLas are human, so we're going to download, go to the FASTA sequence database, and we're going to download human. And we're going to add the IRT sequences because we do have IRT sequences in there. Those are synthetic peptides that can be used for quality control, which I added to these. And so we're going to go human, reviewed sequences only, add common contaminants, add decoys, add IRT sequences. And then we're going to hit OK. And we're going to give it a directory. I like to put these somewhere you can remember. Analyze your own data. All right, and we'll give them a new. We'll just leave it here. Select. It'll download it. Only take a couple minutes. Should anyway. Yesterday it was kind of long. I think Unipro was having issues, but we'll see. We'll see how long it takes today. And just a reminder, if you want to do another organism, you can find these numbers. Prot. Let's go to Uniprot. And you go species. And let's say we were looking at something like E. coli. That would be UP000625. And again, we go to Uniprot and look at species. And then all you have to do, you can see it's done. All right, awesome. So that's done. And then let's say you wanted to download a different, search it against E. coli. All you'd have to do is hit other, and then put in that UP number and it would download it. So be careful with other organisms if you do reviewed or non-reviewed, because that can make a big difference, because some organisms don't have a lot of reviewed sequences. So for example, let's say we were looking at, these are the big ones. So let's say we're looking at cows. Let's see, cows might be a good one. Let's see, and we go to number of entries. And you can see there's only 6,000 cow sequences and 51,000 unreviewed. So there's 6,000 reviewed, 51,000 unreviewed. So be very careful. So if we were to download cows sequences, and this would be, let's see, it won't let me open two of these at once. This would be UP 9136, download, we go other 9136, and we don't want to do reviewed only because it would only download 6,000 sequences and then you would be screwed. so you can uncheck this if we were to search for cows, but since we already downloaded human, we will. Let's see, MS Fragger, we're going to leave all other default things the same. We're not going to change any of these. PTMs, all of this will leave the same. Don't worry about it. Don't worry about it. Don't worry about it. Skyline, we're going to generate a Skyline document. And we're going to use Skyline daily. And make sure you download Skyline daily. To find Skyline, you go skyline.skyline.ms. And then for the daily version, you're going to have to create an account. So you have to go sign in and create an account. And then you go to Skyline and download daily. And then, let's see, sign in to access the Skyline daily download link. So sign in, boom, download it, install it. It should work out of the box with Fragpipe 22. It doesn't with the new version of Diane, which we'll get to in another video. But for this one, I believe it does. All right, so we'll want to check generate Skyline document. Leave that all the same. I don't know why you'd want to change that. Maybe if you're working with modification with Glyco, you can do that, which looks pretty cool. All right, so because the dia tracer takes a long ass time, we are going to start it now, and then I'm going to come back, I'm going to pause the video, and we'll keep going. So let's tell the output where we want the output. And I put this in my directory, I think, analyze your own data, and we're going to give it a new folder and call it Fragpipe. So Fragpipe generates a ton of files, so we're going to say put it all in Fragpipe, hit select, and then we're going to hit run, and it should work. And then through the magic of videos, I'm going to pause it and then come back in like 2 hours. Oh, fast path. Okay, so let's see. So it gave us an error about the database. And why did it give us an error? Oh, because I'm an idiot. And so it doesn't like spaces. So make sure, did I download it here? Okay, so I accidentally downloaded it to a folder with the space in it. Fragpipe doesn't like that, so I'm going to recopy it here, and then just hit browse, and then go to the Fragpipe directory and select that. And now it doesn't have any spaces, so make sure there are no spaces in any of your file path. Then I'm going to hit run, and it's going to give you a warning because it's going to say, hey, there are files in here. But the only file is the FASTA file, which Fragpipe lists as an FAS file and not a FASTA file, but this is the exact same thing. All right, do you want to proceed? Yes. Boom. All right, I'll see you guys in like 2 hours, which will be like 2 seconds for you guys. All right, see you in a sec. Okay, is that working? I think it's working. All right, everybody, we're back. Why is this? Minimize this. So where we left off, we were doing the newest version of Fragpipe, version 22. We went over where to download it, how to get it all running, how to add your files, how to tell the software it's DIA. Even though this is timsTOF, I just noticed we didn't check the ion mobility MS data. I don't think, as far as I know, you don't have to. I checked with the authors of the software, and it'll automatically know it's ion mobility if it's timsTOF, so you don't have to check it. So I don't think we screwed that up. And just to let you know, we did the new DIA tracer, which does diaPASEF data. We downloaded a human database. We had it output a skyline workflow, generate skyline documents. So we'll be able to visualize the data, and let's see if it's done. So it analyzed these 8 files in 194 minutes. So it took a long time to do it. But anyway, that's how long it took. And let's look at our data. So the first thing we want to do is look at the output data for this. And where did I put it? Under FBRET, analyze your own data. So let's see here. FBRET, analyze your own data, Fragpipe. Okay, so you see a ton of files in here. There's the Diane output, because it uses Diane to search it. And this has a very good report PDF, which you can look at. I like looking at that. And you can see various things about your identifications, total quantity, MS1 signal, MS2 signal. So these are probably the 200 nanograms and the 50, so you would expect less signal there. Anyway, you can look at those. Those are kind of useful. In order to look at how many proteins and peptides we identified in this data set, and there's a ton of files over here. I believe the easiest way is to look at, let's see, where did it go? I think it is filter. I don't think it's filter log, is it? Let's see. Yeah, there it is. Okay, so you can open a file called filter log in your Fragpipe output, and this will tell you it identified 18,000 PSMs, which are peptide spectral matches, 15,000 peptides, 2,800 proteins. I think you want to look at that one, right? 2,800 proteins. Let's see, 19,000 peptides, 17,000 peptides. I believe you want to look at the one, most likely the one on the bottom. So 17,000 peptides, 18,000 PSMs, 2,800 proteins. All right. So now, where are our peptides and proteins? So you can open the protein.tsv file, and this will list all your proteins here. This will list a bunch of various statistics like top peptide probability, total peptides per protein, unique peptides, razor peptides. I think I have another video on razor peptides. Total spectral counts, unique spectral counts, that kind of stuff. Let's see. We can filter these and look at sort by total peptides to give us kind of a quasi ranking of abundance. It's not that accurate, but it's okay. I kind of like doing it. All right, so you can see all your info on all your 2,800 proteins, 2,801, because we have one header up at the top. Okay, let's not save that. If you want to look at your peptides, you could do that here. Let's see. And here are your peptides. It'll tell you what proteins or protein groups they're in. And remember, a protein group proteins, because a peptide can be in multiple proteins. And if it is, you'll see that here because there'll be multiple entries. I don't see a lot here. It'll show you the gene name, the ID, observed modifications, assigned modifications. Let's see where in the protein this is, peptide length, that kind of thing. Okay, that's really interesting. All right, don't save that. All right, so what can we do with this data? You can also see the proteins in the dian output, and that would be report protein group matrix TSV. And you can also see the report stats. So there should be 2,800, and there's not. There's 2,762. Why they're a little different, I don't know. We'll have to look at this later, probably because it's a protein group. You can see the quantitation, the expression values for all of these proteins. Okay. And here is the report stats. Okay. Convert. And this is the number of proteins we identified per file, 2,600 approximately, and it gives you like total signal, that kind of thing. And let's see here. This is very interesting. I haven't looked at this in great detail since it was released, because I don't use this a lot. I usually use Spectronaut. Okay, so what else can we do? We can look at, oh, there's a bunch of like files here for retention time alignment and stuff. You can kind of ignore those. There's the skyline output. We can look at that in a minute. It looked like that worked. There's a open Fragpipe PDV viewer. I have no idea if this will work because it's the DIA data set. Let's see. That'd be interesting. Maybe this is what, maybe this is the Spectra DIA tracer does. Let's see. So I believe what's going on here is DIA tracer will make pseudo MS2 from the, deconvolute it from the complex DIA spectra. And remember, DIA spectra contain dozens of MS2 peptides that are overlapping, and then I believe it searches that with MS Fragger, validates it using MS Booster, and then makes a spectral library from that. That spectral library is then used by DIANN to search, and then you can visualize the whole thing in Skyline. So if I'm right about this, spectrum, invalid spectrum path, I don't know what that means. Let's see. Okay, so here, maybe you can see it doesn't seem to be working there. Invalid spectrum path. Reading spectrum file. I don't know, this doesn't seem to be working. I don't know why. It might have been interesting to see the pseudo MS2 spectra, but it's not really my priority. I don't know why it's doing that. Did I break it? I broke it. Oh, let's see what it does. Okay, so while it's busy choking and doing that, let's look at, we can also look at Fragpipe Analyst. And this can show us some interesting figures about our 50 nanogram and 200 nanogram results. So let's see, we want to do LFQ, I think is what we want to do. Upload combined protein.tsv. So we are going to want to do that here. That should be under the Fragpipe. I don't think there is a combined protein.tsv. I think there's, maybe we'll just do protein.tsv because I don't see a combined protein.tsv. Let's try that, see what happens. Okay. Upload sample annotation, and I believe we want to use experiment annotation, I think. Is that right? And we can use Bax LFQ intensity. I believe that's what Diane's using anyway. Now let's see if that works. not work. Combined protein. So maybe we don't use protein. Maybe diane output. Maybe you want to use the diane output. PSM protein peptide library. Use no. I don't know. It doesn't look like that's working for diane for this. Maybe let's see. I'm gonna do. Let's see if it works with the diane output. So if we do like report protein groups matrix, maybe that'll work. Probably not. No, I don't think that'll work either. Anyway, I don't think this works. So maybe it only works for DDA. It doesn't work for the DIA data sets yet, so we can ignore that. But let's look at the skyline output. And what we do is we go and open Skyline daily, which we installed last time, remember? And there's an upgrade, or an update, because it is a daily daily thing. But let's just remind me later. Okay, so we're gonna want to open it. But the easiest way to probably open it, let's see, is Fragpipe. Let's see. Skyline output is just to open the fragpipe.sky. Let's open that. It'll be in the Fragpipe Skyline output. And it looks like it's working. Awesome. Okay, there we go. Peak area replicates. We'll move this around. Skyline is very pretty. Okay. Peak area. Awesome. And here's our IRT kits. Our IRT peptides that should be in the same amount in every file, but it doesn't look like it really is. There's something wrong with this one. I don't really know why. Is that consistent? Looks like it's pretty consistent. But what's really nice, so either I pipetted that wrong or something else happened. So here you can see the Skyline data for these transitions. These are on the upper, these are the precursors, so these are the intact peptides. These are the Y and B ions, so the fragment peptides from this intact precursor. And you can see those here. You can see those here. You can see what fragments it's using. It's using all of them. It's kind of weird. All right, so we can look at any of our 2000 proteins. You can see some look better than others. You can see what fragments it's using. Seems like it's using an awful lot. I don't know if there's a way to change that. Like this one's only using 1, 2, 3, 4. That doesn't look very good. This might be a good one to check with Spectronaut. All right, let's do that. So anyway, this is the kind of visualization data. You can see this ID is looking kind of sketchy. Not much here on the precursor. You can see a couple things that are co-eluding. But if you zoom out, you can see, whoa. That doesn't look great. But is it real? I don't know. Let's go check. All right, so let's see what Spectronaut showed from the same data set. And I have that here. This is the same data set we searched with Fragpipe that we searched with Spectronaut. You can see, right away the number of IDs is almost doubled. So Fragpipe implementation of diane, the way it does DIA tracer and uses Fragpipe and gets pseudo MS2 spectra, identified about 2,800 protein groups. You could see that Spectronaut identified 4,400, so almost, not double, but close. Why is that? I have some suspicions, but I don't know. But let's look at some of the same peptides and see if we could match them. So you can see on Spectronaut, you might get this too if you use some of our Spectronaut data. This will be red. It basically can't find the raw files, so we can look for them. Click these 3 dots and say map missing runs, and then you can look for them, and I think I have them. You can either give them the converted HTRML or the .d files, and we'll just do that. And it doesn't, and because I use the HTRMS files and I don't have those, it's not going to let me look for it. It's not going to find them. So Anyway, maybe we can still, it looks like we can still see the data, so that's nice. So let's look at this peptide here, and we can hit copy, and let's see if we can see it in Spectronaut. So in Spectronaut, if you want to find a peptide, you go here, and here's these filters, and you go to assay, and you hit peptide sequence, and there's a ton of stuff like this. And you paste in the peptide and delete all this other crap, because it can't find that. And we hit OK, and it found it. All right, so in order to match the correct one, we're going to have to figure out what file this is from, because we have 8 of them. In order to figure this out, let's look at, this is 6367, 6368, 6369. And so you can see all of these here. They're all equally kind of crappy. So let's look at 6367. This is what our Fragpipe implementation skyline data looks like. And skyline is very pretty. And let's see what our Spectronaut looks like. And we go to file, make sure we're in the same file. 6367, which is right here. Here's our peptide, and there it is. Let's see if I can put these in the same pane. Okay, so 9 minutes, 300ths of a minute. Oh, there it is there. There it is there. It looks a little better here, and let me show you why it looks better here. All right, so let's look at what fragment ions it's using. It's using Y9, 7, Y5, and Y11. Let's see what these are using, and you can see the precursor is the same as it should be. It's 709.3935, and it is using, let's see if it's using the same ones, and it's not. So this is the secret to DIA, picking these right fragments, right? So like there might be a good example in the spectral, no, not in the spectral library. However, this one has, remember there's an overlapping ladder series of Y and B ions, so there's a ton of them. And I think Skyline makes it easy to see all of them. And let's see if we go here. Let's see if we can filter all of them. And you could see there's a ton of them, right? We only see the Y and B ions, but there's a ton of B ions and a ton of theoretical Y ions and theoretical B ions that you can choose, and you will not see all of them, right? Which ones do you choose? You choose the ones, usually the deep learning predicted software says to choose. Okay, so Spectronaut is using Y8, Y9, and Y3. And this is using Y9, Y7, Y5, and Y11, so completely different. And the ones Spectronaut are actually using for quantitation are blue. The ones that are not blue, it may have used to identify the peptide, but maybe not quantify it. And remember, in my previous videos, quantifying and identifying something are not the same thing. So you can identify things with fragment ions that are not good for quantitation. So Spectronaut seems to do a really, really, really good job at figuring out which ones it should use. And I think I even am using the new deep learning timsTOF. I'm not. Deep quant correction. So this is a new feature in Spectronaut 19. You could turn it on with timsTOF data. It uses another deep learning model to try to predict, I think, what fragments to use. I don't think it actually changes for these. I've tried it earlier, but I don't want to reprocess the data, even though it'll take a couple minutes. Let's find another one. This is kind of fun. Let's like randomly pick one. How about this one? Oh, that looks kind of crappy. These don't look very good. That looks good. This just looks like semi-crappy, right? So let's look at this one. So we'll copy it. We'll paste it here. Let's see, delete all this other crap and see if we find it. Let's see, so GIGMN, and this is 6367, 5 profile 6367. I really like that on the timsTOF, how each run will give you a different. There we go. Boom. And it has a carboxyamino methyl, which you can see here by an underlined C. 7 minutes. This is what it looks like here. Oh man, look at these. So these are using a lot of fragments. Why it's using like all of them, I don't know. Like, I really don't know. This is what the pseudo MS2 spectra look like from diaTracer. Okay. All right, so it's using a lot of fragments. And this one is using, boom, it's only using 3 again. So it's picking probably the best 3 that align across the different runs. So let's look at this one. So we can pull this out here and look at this peptide peak area across our different runs. And I believe these are all 200s, and then these are 50s, so you'd expect them to be less. We're not normalizing. I believe I have normalization turned off here. I do not. So why don't we do reprocess the data, because this is in another video. So let's turn that off and let's turn on deep quant and hit confirm. And you guys can do this in your viewer too. Okay. Come on, light. Turn on. Thank you. All right, that only took a minute. This will probably close again, so I'm going to copy this before it disappears. Let's use B6, Y10. And I knew it would disappear, so we're going to go assay, peptide sequence. Go back, there, 6367. Looks exactly the same, I believe. Yes. We can go back to the video to see if it's using the same ones. I don't know. I've already forgotten. Maybe it's not. Let's rewind the video. We can rewind the video and see. But what we want to do is, because this isn't normalized, we should see less peak in area on the 50s than we should on the 200s. And let's see if we do. A good assay for this is we can look at a cross runs peptide versus precursor. And you could see we do. So there's the precursor versus peptide. And you can see there's a lot more. There should be 4 times as more. It doesn't look like there is in some of them, but it could be pipetting errors or something. You can kind of see here. So anyway, less, as you expect. So that's good. I really like the skyline visualization. It's very, very pretty. Okay, so you can move windows around, dock them places. Here. Let's see. So you can see they're kind of 7 minutes. 7 minutes. Found the same thing. So skyline. So the Fragpipe skyline dian implementation identified about half of what we are with Spectronaut. Why that is, I don't know. But anyway, Spectronaut's not free. Fragpipe and Skyline are. So if you don't want to pay me to process your stuff in Spectronaut, you can use this video to show you how to do it for free. I personally don't like this implementation, and I like to use dian by itself. So in the next video, I'm going to process this again by dian, and it also has Skyline built in, and we're going to compare it with the Skyline from Fragpipe and the Skyline from, and look at the Lucian group profiles from Spectronaut. So anyway, that's it for Fragpipe 22. You can analyze your own data for free. You don't have to pay me anything. It's fun to learn how to do. Their Fragpipe support is excellent, and Feng Chao will get back to you with like usually in minutes if you find something wrong. And one of the advantages of doing it this way is you can look for PTMs. I think a lot easier. I don't know if it'll work with open PTMs, but they have a nice phosphorylation one. So maybe if you have a phospho-enriched set, you can look that up. Okay, all right, thanks everybody. Next video will be Fragpipe, will be dian, the new version of dian that also has a Skyline viewer. So we'll see how that looks too, and we'll look at some of the same peptides. All right. Bye, everybody.